Good morning, everybody, and welcome to Osher. Osher Lifelong Learning at UCSD. I'm Mavis Porter. I'm a member of the Curriculum Committee. First of all, I'd like to begin by thanking Carta for bringing the 5 excellent lectures to Osher, and also I'd like to thank them for their great research. In particular, I would like to thank Lindsay Hunter. She just did everything to bring this thing together, as far as I'm concerned. I'm here to blab my mouth. But anyway, today, as you know, our speaker is Professor Paul Gagneux. Professor Pascal Gagneux is a professor of pathology and anthropogeny at UCSD San Diego, and he is an associate director of Carta. He received his PhD in zoology at the University of Basel in Switzerland. His research focuses on the evolution of primate molecular diversity and how it is shaped by reproduction and infection. I'm assuming when we're talking about primates, we will be included. Anyway, welcome, Professor.
Thank you very much for the kind introduction. Thank you all for being here. I'm going to remove my mask if that is okay with you, because the accent is enough. We don't need muffling on top of it. So I have a lot on my plate today, and I'm going to go relatively fast. Bear with me, because I want to try to combine a very brief introduction into anthropogeny with a discussion of emerging diseases and the concept of One Health. We are the planetary species. Humans are the only primate that now live on all continents, and you can see this as beautifully illustrated with an image produced by technology that humans collaboratively have produced, satellites that can image what happens at night on the planet. You can see the entire world lit up, especially where people have more money. In contrast, the great apes are restricted to the forest to tropical forests. And that is even the fact for our 2 closest living relatives, chimpanzees and bonobos, that are, based on DNA comparisons, they are more related to humans than they are to the other great apes. So there's something odd about this great ape with minimal hair that has conquered the planet. And there's only one other species that does that. It's not a primate, it's the orcas. There's only one species of orcas, and they are periplanetary, restricted to oceans, of course. They have very big brains, and they have grandmothers. Hold that thought. So if you were to depict what humans do that is odd, the list is very long, but one of them is this is 1 day's worth of intercontinental flights. Very important for the concept of emerging diseases, because a virus in the forest in the Congo today can be inside the vein of an an IV drug-using person in Washington or Paris or Zurich the next day. In contrast, and actually COVID has really reduced this, but it's now coming back very quickly. In contrast, our closest living relative, chimpanzees, jealously guard a territory about the size of Mission Bay and Pacific Beach, and have zero tolerance for foreigners, especially foreign males. The other closely related species, bonobos, are a bit more tolerant. These are actually 2 social groups that briefly meet, exchange a few genital massages, not an orgy, but like hugs and kisses, and that allows them not to kill each other. But overall, they also live on a territory about the size of Mission Bay and Pacific Beach, and do not roam the planet like we do. This is my colleague Sarah Bleffer-Hurdy, who is a very famous primatologist, has come up with this thought experiment. Imagine 300 unrelated male chimpanzees on a jet airplane and fly them from LAX to Zurich. It makes us feel so good, because aren't we civilized and socially tolerant? Unfortunately, we do things like that with airplanes, right? So we are extreme in all directions, the good and the really, really bad. And the list doesn't end there. You can pick up a dictionary or an encyclopedia and look at astronavigation, beekeeping, bicycling, X-ray, yoga, or ziplining. You can train some chimpanzees to do some of these things, but it would be humans who give them the technology and the training to do them. So the list of what we do is endless, that only humans do. You can see a chimpanzee down there, aptly named Darwin. He was actually the first wild chimpanzee I met many, many years ago, 35 years ago. He's cracking nuts with a hammer. It took him 5 years to learn this, and there is archaeological evidence showing that chimpanzees in that part of West Africa have been cracking nuts with hammer for thousands of years. The hammers haven't changed. When the hammers break, they abandon them and look for a new one. Humans, on the other hand, have this cumulative culture where we can make simple Aldoan tools, then we can improve them, make them symmetrical, then we realize the chips are the best part, we can haft them on arrows, we can invent new stones like pottery, we can make stone, translucent stone out of sand when we invented glass, and then photovoltaic panels and microchips and so on. So humans build on ideas of others, and that's what you don't see in non-human animals. Even though many non-human animals have material cultures, but they don't have this cumulative culture where we build on ideas of others. But more important than the material culture is what we can do with our interpretation. I hope you see the footprint here. There's a whole bunch of footprints. The first big one in the red circle is actually a human. And the arrow, this is the other business end of an arrow with the fletching, is held by a Hadza friend of mine named Sikwezi, who points out this footprint that I didn't see. And he says, this is Philemon, and it looks like he hunted 2 zebras, based just on footprints, right? And then it goes on, it says, actually, after that, the toga pastoralist in sandals came by with his cows. There is no molecule there. There is nothing there that is directly connected to these animals. It's the beginning of symbolism, and people like Louis Liebenberg have actually caught our attention to the fact that no other species is known to do that. I've queried all my primatologist colleagues and asked them, have you ever seen a great ape getting the footprint of other great apes? Said, no, the closest I got was a chimpanzee who looked at a knuckle print of another chimpanzee, and he had rained, and he must have seen his reflection because he jumped back and it scared him. So this is incredible. Why we are the only species? Your dog is really good at tracking, but it's a molecule of whatever the dog is tracking that hits the receptors in the nose of the dog. There is no molecules here. Right? This is the beginning of symbolism and getting signs. And actually, let's go to infectious disease. We now use this to detect the footprints left by the spike glycoprotein of COVID on our immune system. When you do an antibody test against COVID, that's what you're doing. The virus can be gone, but the antibodies that formed by your B cells have actually the imprint of the virus. And in cyan or in pink here, you can see the really important antigenic epitopes that are important to track here in the case of the Omicron strain that I had the pleasure of having this July while I was in Tanzania. So humans have become symbolic. These symbols are symbols for us. We can refer to each other in absence of our friends or enemies. And we can use these terms to build entire kinship terms and tribes. We can band together. We can have shared rituals, dance, music, sacred spaces, group identity, shared practices, very importantly, cuisine that defines us. The last thing that is lost by different cultures is usually their cuisine. Lost the language, the religion, the dress habit, but you still, go and try to find the food. And then importantly, we have evolved the capacity to use these markers of group belonging to cooperate together in huge groups of groups. And ironically, this cooperation very often is in order to compete with other such groups of groups. So in a way, humans, the only primate that has this doing so, has reinvented what eusocial insects such as ants. They have culturally reinvented group markings that in the ants are actually chemicals, simple chemicals that tell a big group of Argentine ants in San Diego, for example, to tolerate the other Argentine ants. And humans have changed something in their life history, which is the term biologists use to refer to the schedule of life. Are you born alone? How long do you take to sexually mature? When do you start reproducing? And when do you stop reproducing? And are 2 periods of the human life history that dramatically change, that are unique to humans among primates. And they include a long childhood where body growth, somatic growth, is slowed down, but the brain continues to grow at a huge rate, taking in all kinds of inputs from nursery rhymes to school songs and so forth. And then almost 25 to 30% of longevity in females is non-reproductive because the ovaries are done, no more ovulation. Very fit, about 25% of surviving adults in many hunter-gatherer groups are postmenopausal females who play a very important role in transmission of ideas and help with producing children. So the irony is we have these helpless children with huge brains that grow very, very slowly, need a lot of help. And yet humans can reproduce much faster than any of the great apes. How do they do it? Almost certainly with help from non-moms, mostly grandmothers and aunties and so forth. So that's just the data on 5 wild chimpanzee groups. You see the age structure. There's a few that survive the end of making eggs. And in humans, it's actually quite a large fraction. And these ladies, just my friend Heidi here, who's using my Swiss pocket knife to cut a huge tubers, she just dug out with a simple wooden stick. And you don't see this in almost no other species. You see it in hunter-gatherer societies, and you see it in orcas. They come back. But that's it. We don't know of any other animals that have a large proportion of their population as grandmothers. We know now, based on archaeological and DNA data, that our species, Homo sapiens, took over the planet in the last 100 years, and the Americas were the last 2 continents where they got to. Actually, genetic data would indicate it was 15,000 years ago, but there was a problem because we have archaeological data that puts modern humans in North America and in South America as early as almost 30,000 years ago. So stay tuned for some fine-tuning, but overall that's the picture, right? A new species out of Africa in the last 100,000 years. Now, ecologists have a way to take existing data on ecosystem, plants, and climate, and predict based on the plants you see, how many animals would you expect, like megafauna, big animals that are 100 pounds. If they do so, the color, the color map at the bottom, it's like a heat map. The warmer the color, the more large animals you expect, mostly mammals such as gazelles and elk and elephants and so forth. So the bottom one is what you would predict based on current climatic and ecological information. And the top one is what you observe. The only large herds of animals you can still see are in East Africa and South Africa. Why? What happened? What happened is us. So this is in Ngorongoro crater, a place the size of San Diego with 22,000 large vertebrates that you can still go and see. Everywhere else, when modern Homo sapiens arrived, there was a mass extinction event. So we've started overhunting well before we became mechanized and we invented steam engines and combustion engines. And of course, these combustion engines and steam engines are what started driving the climate change. Temperature has dramatically started rising, measured as the Keeling curve on Mauna Loa in Hawaii that is now interrupted because of the eruption. This period of relative warm before the spike of the Keeling curve is known as the last interglacial. And the last interglacial was important for many reasons. One of them was that it's the beginning of the 6th mass extinction at the hands of us, modern Homo sapiens. You can see it more here. It also corresponds to the invention of farming in at least 9 different places around the world, from Papua New Guinea to the Indus Valley to the Huanghe Valley, to the Levant and the Fertile Crescent, to West Africa, to Central, South, and North America, all places that have independently invented farming. Farming was wonderful for civilizations, but it brought within the so-called 5 Ps: poverty, poor health, plunder, politics, and power differentials. And I would suggest, it was based on domestication of plants and animals. The first animal to be domesticated were dogs 30,000 years ago. After that, it was the pig. There's some new data on geese very early on. And they brought with them 2 additional Ps, namely many new pathogens and parasites directly taken up from these species that we humans lived in very close contact. To illustrate this, you see a photograph of this wildebeest, again in Ngorongoro crater. There's about 5 million large mammals in Tanzania. And Tanzania makes more money from tourism for safari than it makes from gold, and it's one of the biggest gold exporters. But there are 33 million cows in Tanzania as well. So we are now 8 billion people, just crossed that threshold last week, and there is more than 1 billion cattle that live on the planet. It is possible to learn from people who are pastoralists, such as the Maasai, about conservation. And if you're interested in this topic, I highly recommend this book by my colleague David Weston, also a CARTA member, called We Alone: How Humans Have Conquered the Planet and Can Also Save It, based on the observations that the pastoralists, the Maasai in Kenya, have a profound way of actually protecting wildlife that they consider emergency meat if the cattle die off. So we've also farmed, planted a lot of plants. Essentially, you can think of us as a grass-eating primate The 4 major crops on the planet in all civilizations are based on starches. We now mostly grow 3 grasses and 1 bean: corn, rice, wheat, and soybean. And that has propelled us into 8 billion people. Half of us live in cities. And the more people live in cities, the less they know about natural processes, ecosystem, and the less they are oblivious of the fact that natural systems are full of pathogens and parasites that can jump between species. And those people living in the cities have a huge footprint on the planet because they demand a lot of goods. They are interested in status. That is often what brings people to cities. To produce all these things, they need primary products, such as timber and iron ores and so forth. This is a map of the Congo that shows you next to it a photograph of a brand new logging road. Look at the red lines in the Congo there. This gigantic place, Congo. You can see the density of logging roads. And these logging roads go into the very heart of the Congo basin. Once you put a road in like that, the poorest people who have no chance in the villages or in towns might go along these roads and start putting up little villages. And when they're hungry, they hunt. And when they hunt, they sample everything that lives there and all the parasites and pathogens they have. So this process of driving deeper into the Amazon basin, the Congo basin, and some of the large forested areas in Southeast Asia brings people into new contact with more species at a rate that has never existed before. And that is all driven by our demand. Just to remind you, by virtue of living here in San Diego, you use about 100 times more energy than a Tanzanian every day, even if you ride your bike like I do. And you produce tons of trash. This is Miramar landfill. It's illegal. After I took that photograph, they said, no, you can't take photographs. And a lot of our trash ends up in the Pacific, in the mid-Pacific garbage gyre, right? So huge problem. So hopefully we recycle. One of the things we recycle is tires. When we do so, the tires can be rained on in transit, and then mosquitoes hatch in them. In 1985, Aedes albopictus, or the tiger mosquito, arrived in Houston, Texas, courtesy of recycled tires from Japan. It is now firmly established here and is awaiting chikungunya virus, West Nile virus, a lot of other nasty things, even yellow fever that it can transmit. Talking about yellow fever is an African virus, but there's a very good vaccine, lifelong immunity with 1 shot. It's phenomenal. I highly recommend it should you go to the tropics. Anybody knows how yellow fever came to the Americas? With the transatlantic slave trade. How did the mosquitoes come to America? With the transatlantic slave trade in the freshwater drinking barrels on these absolutely horrific passage boats. And now it infects a lot of primates in the New World and is established. Right? And was very important actually in American history. All the way to Philadelphia, there were yellow fever outbreaks. But as I said, we're lucky we have a really good vaccine against this. So a few words about pathogens. Pathogens are invisible, which is why all traditional systems of healing do not have a concept for viruses, including Western medicine. Right? It's only 19th century discovery. There was something smaller than bacteria that you can press through a filter and still infect. So viruses are, many of us have learned much more about viruses recently because of the pandemic. Protozoans, which are actually tiny little animals, single-celled animals, such as malaria. Bacteria, also not visible by the naked eye. Helminths, some of those you can actually see, including tapeworms, but there's filaria that are microscopic. And last but not least, prions, things that cause Creutzfeldt-Jakob's disease or Kuru or mad cow disease that is also massively amplified by agricultural practices. So the notion of emerging disease has been around for several decades, and you can note the last author on this paper was Tony Fauci. This was from 2004. It was a beginning overview of where around the world which pathogens pose a distinct and clear danger to jump into novel human populations and potentially kickstart a new pandemic. And that now with the power of being able to sequence DNA and RNA, you can directly sample viruses in everything, in soil, in water, in everywhere you look. And so the estimate is that there is more than 500,000 viruses we don't even know about. So the options for new viral infections are endless, right? It will never end. Some of the diseases are very old. This is the story of TB. Long in the textbook as something that the cows gave us. It's not true. Actually, TB came out of Africa with humans. So you can retrace the out of Africa movement of Homo sapiens based on the DNA of Mycobacterium tuberculosis. And after that, during colonial expansion, you get a second wave of TB that goes wherever the Europeans arrive and colonize. Interestingly, this might be very old and connected to the use of fire. What does fire do? It brings us together every day. We talk to each other. It damages our lungs. Meanwhile, we have perfect opportunity to exchange infections. So very interesting ideas of ancient coevolution of human-specific disease and fire use. Jump to now. Hopefully you all got the flu vaccine. Influenza A is an RNA virus, very much like corona, but a bit more nifty because it has a segmented genome. It has different little chromosomes that it can exchange with co-infects, so it can recombine and create diversity. Influenza virus is a bird virus that jumps into humans every year and can also jump in pigs and then mix in pigs and birds and cause pretty nasty disease. The picture is complicated. We will never get rid of influenza viruses. There is an endless pool of these viruses in aquatic birds, and some of these birds migrate from pole to pole every year. Very, very mobile, and wherever they poop, they deposit RNA virus that can then be picked up by other birds or by pigs or by humans. And you all are familiar with the Spanish flu or the massive influenza pandemic that actually killed fewer Americans than COVID has, right? COVID has now killed 1 million Americans. It was a much larger fraction of the American population back then. But it killed about 50 million people worldwide. And so this is the horrific story of massive pandemics of the past, and we have to update it, right? There were things like Ebola in 2013 in West Africa, Zika, where we still don't have very clear data. I'll talk about Zika briefly, and then COVID, that is now over 6 million deaths and over 1 million of them in the US. I want to briefly take you into a cell, because that's what a virus does. A virus gets into the cell and then tells the cell to make new viral particles. So it's a parasite of cells. And you've always heard that the genome is the blueprint of life, right? It turns out it's more complicated than that. Yes, the hereditary information, the DNA or the RNA, is found in the nucleus, but it then is expressed and goes. RNA is made from DNA, the RNA goes to ribosomes, and then proteins are made in these little vesicles that travel towards the surface of the cell. These proteins are altered, sugars are added to them, lipids are added to them, and then they come out and they form the outside of the cell, or they are secreted. So living cells are a gemisch of a lot of different classes of molecules, and not all of these are in the genome. Right? Molecules come from the food, such as sugars and fats, and together they form something much more complicated than a blueprint for an architect who builds a house. So by the time you make different cells, for example, we all started out as a sperm and an egg meeting. If you home in on the surface of different cells, a sperm, a red blood cell, a leukocyte that will make antibodies or eat up something scary, or an epithelium, which is the first line of landing of any virus or bacterium in your lungs or in your gut. These surfaces have different flavors. It's as if they each have a different molecular ecosystem. Right? We're now 1 million times smaller than we are. So it's a very small world, but it's hugely diverse. And these different ecosystems, molecular ecosystems, they're very important. They decide that a COVID virus is really good at getting into your lung, or a polio virus has evolved to look for the landscape on the epithelium of your gut only. That's where it infects. So there's a lot of interest, and my particular interest is how different diseases are pushing these landscapes by trying to get in. And of course, we have to change them and protect the landscapes so that pathogens don't come in and kill us. And interestingly, I mentioned this here, you can see at the end of many of these chains are little purple diamonds. These little purple diamonds are a sugar known as sialic acid, which I'm very interested in. I will be talking about it a little bit because it involves some very interesting human biology that defines us, even compared to our great ape relatives. So this molecule at the very tip of these forests is like a flavor of self. It is used by our innate immune system to recognize self. Even though you're a different cell, you're still a human cell, a healthy human cell. You don't have cancer and you're not infected. So think of this molecular landscape. I'm taking you here to West Africa. This is a canopy of rainforest. And that rainforest is defined. You now look at The ground of the forest would be the membrane of the cell, and what comes out are proteins and glycolipids that have these chains of oligosaccharides, and the last ones are these little diamonds called sialic acids. And because they're out there, they are what viruses such as influenza A bump into. You've all heard H1N1, H2, N3. H stands for hemagglutinin, which is a protein that can grab sialic acid, and N stands for neuraminidase, which is the other protein that can cut sialic acid. So influenza viruses have something to grab these sugars and something to cut them off. Why would this be? Because they can eat their way through your secreted mucins and get to your cell surfaces. A much better analogy for these surfaces on your cells is actually the kelp forest from right out here. And kelp forests they move in the water. Same thing for these molecular forests on your cells. They are hydrated and they move. And if you go to Northern California, you see this bull kelp that has all these little bubbles at the end. Think of those as sialic acids. And if you're the virus, the analogy would be you'd be in your kayak and you hold on to one of these things, and that would be the influenza virus or many of the other viruses that use sialic acids to get into cells. So here's the problem we face, right? We are multicellular, long-lived animals that, if we're lucky, live 80, 90 years or even more. Our cells are covered with these, I've really simplified the forest here. All I show you in red are these little red diamonds, which are the last sugar on the surface. So that's how our cells talk to each other. We have these sugars on every one cell, and then we have these bubbly things that are called receptors, or they're proteins. They can they can check out the sugar on the other cell and say, yeah, that looks like it's hunky-dory, it's healthy, no problem. In come the parasites, things like malaria. They recognize sialic acid. That's how they get into your red blood cells. Viruses recognize sialic acid. Bacteria also recognize sialic acid. A lot of E. coli strains will specifically grab on. Worse, these little guys, they evolve 1 million times faster than us. They have cracked the system. They can put sialic acids on themselves. They either make them themselves, or like a decorator crab, they take them off you and put them on themselves. Now we have to detect bad little guys that want to kill us, that look like us. How should we do that? That's a problem. One of the ways we can do that is we can evolve new grabbers, new little sensors that will recognize those sialic acids and then tell our system, our immune cells, hey, this doesn't look kosher. It looks almost kosher, but it's not kosher. Better do something. And there are examples for that. So it's a coevolutionary arms race between the molecules on our cells and immune detection and the many ways that parasites and pathogens use gimmickry and mimicry to hack our systems. So maybe not surprising, there are certain events that have happened, and this is a really famous one that brought me to UCSD and then caused me to stay here for the next 30 years. It's the first known biochemical difference between humans and other mammals, and this was discovered by my colleague Ajit Varki. And it's a mutation that destroyed the function of an enzyme that happened about 2 million years ago, and what happened is that it caused humans to be incapable to modify one of these surface sugars. And I will illustrate this for you in just a moment. And we were able to time it. I was actually involved in trying to time when this happened. This happened about 2 million years ago. So it looks really simple. Forget the structures, just remember that one of the sugar that is on your cells can be modified into a slightly different sugar. So your surface has a slightly different flavor. Chimpanzees can do that, your dogs can do that, your cats can do that. Humans cannot do that because this gene is kaput in humans. We still find the DNA in the genome, but it doesn't work anymore. What this means, I'm illustrating it here at the level of a red blood cell. You see the little red blood cell, like a bowl, like a punch bowl out of clay. These transport gases, right, in your blood that make you turn your blood red. If you landed on a tiny little piece of surface of this cell, that's what the surface would look like. There would be glycoproteins and glycolipids that make this little forest canopy. It looks more like, a saguaro forest in Arizona. And the very tip of many of those have a sialic acid there. So that's why sialic acid gives the flavor of self, the biochemical flavor. Humans would look like what you see on the left-hand side, and most other mammals have a gemisch. They have a mixture of 2 different sialic acids because they can modify the purple one into the blue one. So by virtue of having lost these enzymes, humans have changed how they taste to pathogens. Almost certainly this was driven by pathogens initially. And just to illustrate this, so you can, in a few years ago, I worked on comparisons between human and chimpanzee sperm. We have antibodies that detect these non-human sugars. Look at how bright these chimpanzee sperm are, and look at the human sperm. They don't stain. They also have much smaller outboard engine, but that's a topic for another discussion. So essentially, this proves to you that from the view of an immune system, for example, of a female who mates with a human, these chimpanzee sperm would be highly antigenic. They scream non-self. They do not look like a kosher human sperm. They have the wrong molecular flavor. And when I joined, as a postdoc, joined Ajit Varki's lab, this is a phylogeny representing 5,000 species of mammals. That's about how many mammals there are on the planet. You can see humans down here. Up to 15 years ago, we knew that humans did not have the capacity to make that other sugar. We now know, based on my work and work of other colleagues, that we first showed that the New World primates, which are the monkeys of Central and South America, also cannot make that other sugar. And then people working on influenza showed that ferrets and raccoons and seals couldn't make it. And then people started looking at the genomes of different animals. People working on bats found that Rhinophilus, the bat that almost certainly gave us COVID, also doesn't make that sialic acid. So there's a bunch of other species. Sperm whale, white-tailed deer, cannot make sialic acid for the same reason as humans, but a different mutation, deeper in time, much earlier than humans. So this means that among mammals, there are mammals that taste the same to a pathogen and mammals that taste differently to pathogens. Right? That's a pattern, if you look at evolution, that diversity is protective. Being different from each other can protect you because it can be a firewall to diseases jumping from one animal to the next. Remember influenza A. Look among all the species that influenza A infects, how many of them also lack the non-human sugar? Almost all of them. This is really important. The 2009 swine flu that was first discovered here jumped into northern elephant seals, and it could recombine there and then jump back to us. Right? So some knowledge about the biochemistry of cell surfaces becomes relevant when you try to predict where a disease is likely to jump and where it cannot jump because there is some kind of barrier. A terrible disease that some people have called the biggest killer is smallpox. It's extremely highly transmissible. The good news is it's eradicated. It still exists in some freezers of defense departments, unfortunately. But it has killed so many people over the ages. It came to the Americas and ravaged the populations. It's gone based on a vaccine. And another one is polio, which is not completely gone again and has made a comeback with vaccine hesitancy, so huge problems. But eventually it should go away. Right? You see Pakistan there and Afghanistan, and that's owed to the CIA that used polio vaccination to get to Osama bin Laden. And so that caused a huge step back in trusting people who vaccinate because our vaccine is the CIA trying to get a blood sample. So it's a very difficult thing. I'm pretty glad we got Osama bin Laden, but at a huge cost, right? If you start using public health to do military operations. Now I want to come to monkeypox, which is much less deadly, and it's misnamed, of course, because the host of monkeypox are these squirrels in African forest. Many African forests are so overhunted that people now start hunting whatever moves, hornbills, little squirrels, and then they get monkeypox. And it turns out humans are not the only one who get monkeypox. You can see a young chimpanzee there in West Africa that also got monkeypox. Monkeypox even made it to the US in 2003 via the pet trade with African rodents that were housed together with other animals. But luckily that didn't take off and nobody died. This is a study of monkeypox in the Thai forest in West Africa, where I spent almost 2 years, mostly on that blue circle. You can now use GPS to follow where chimpanzees go. So this shows you where individual chimpanzees have worked, have lived, have moved. And this is from a paper in 2020, predating the latest monkeypox. And the idea is that by keeping an eye on our close relatives and seeing what diseases they get, you might be able to predict the next thing that is going to jump into humans. And so that's the phylogeny studying the DNA of the monkeypox viruses, proving that this is a local infection. None of them died. This is the latest news on monkeypox based on the CDC this month. 82,000 cases. The good news is very low fatalities, but absolutely horrific for the people who got there. Very important to point out, this is very strictly linked to particular behavior that has nothing to do with moral or immoral behavior, but it has to do with very risky sexual behavior, having a lot of male partners. Just like, you know, big wave surfing is very risky, it's not amoral. Having a lot of sexual partner is not amoral, but it's very risky. Right? And that message sometimes didn't get out enough. Good news is it seems under control now. So what is the approach to kind of take care of modern zoonoses? Zoonoses are diseases jumping from animal to humans. Because we now have a planet where people move around the planet almost every day, it really behooves us to keep sentinel systems where we can catch jumps before they spike into a massive pandemic that kills a lot of people. So by having surveillance and have people look at which viruses are around, is there a new virus in a species we haven't seen before, you can prevent both death in humans and death in animals. And this realization that viruses don't necessarily care about the species they infect is what underlies One Health. Having a blind eye only looking at what happens in humans, not realizing that something is starting to rage in animals ready to jump into humans is a huge risk. One example that's still poorly explained is the whole history of HIV. I thought I briefly mentioned that. So 30 species of primates in Africa have their own HIV-like virus, and none of them have HIV/AIDS, the full-blown disease. Humans are an exception. We almost certainly lost HIV, something HIV-like, right? Because we didn't have it until the beginning of the 20th century. How did that happen? We're still not exactly sure. We know roughly when it happened, the beginning of the 20th century. It coincided with one person, King Leopold of Belgium, owning an entire country. Right? The Belgian Congo was his personal possession. There were, guidelines to men from Belgium how to cut off hands of locals to force them to work. And I mean, among the most brutal colonial, if you want colonial brutality, read up about the Belgian Congo. Of course, coinciding with mining, with forced labor, with new cities, with free women who would leave their villages, live in the cities, many of them working as sex workers. Then coinciding with actually blood commerce in Haiti, Haitian help workers that were shipped to the Congo once it became independent. It created a perfect storm for a chimpanzee virus to adapt to humans and then jump via Haiti into the United States and into the rest of the world. You might have heard that there was an infamous person, Gaëtan Dugas, who was a flight attendant, that some people called him the typhoid Mary of HIV or patient zero. A paper in '84 studied his contacts. This is a cluster diagram of him in the middle and 40 sexual contacts. All of them had Kaposi's sarcoma initially, and people didn't really understand AIDS at the time, right? It turns out this is wrong. People now can do molecular sleuthing and sequence the RNA of HIV viruses and prove that he was not patient zero. Yes, he was an amplifier, but he was not patient zero. Right? So the good news is today we have the tools to follow viral epidemics in real time. I'll show you an example with regard to Zika virus. So Zika virus, absolutely terrific, terrible, terrible infection that my colleague Alison Watry, essentially using stem cells, proved that this virus specifically it goes after neural progenitor cells and kills them, which leads to the microcephaly in the children of mothers that are infected with Zika virus. The image you have in the middle here is a phylogeny. Those are based on DNA sequences of Zika virus in real time. So epidemiologists can follow the evolution of this virus. In this case, the good news is that the epidemic is almost over. There's a few sporadic places. But it caused massive harm, right? And these kids are born and they have absolute massive cognitive impairment and they need help for the rest of their lives. They will probably survive their parents, and then who helps them then? We can turn things around. This is from a paper showing that the very chimpanzees I used to study as a field primatologist are dying from respiratory viruses that are closely related to respiratory viruses found in American and European field researchers who are primatologists who want to save chimpanzees. Right? So they're loving them to death. We go there, we spit or we clean our noses, and then an innocuous respiratory virus in us can kill chimpanzees. A few words about bats. Bats are amazing. They're the only mammals with active flight, and it turns out being able to fly actively puts a huge metabolic and immune demand on these animals. So bats have essentially tweaked their immune system to prevent their tissues from melting down when they fly, and in the bargain, they've become totally resistant to a lot of viruses, which now effectively makes them flying viral bags. Right? And they go very far, especially the megabats. This is my colleague Lin Fa Wang, who actually is one of the few people in the world who has a bat vivarium in Singapore. And the way he sold this to the Singapore government is not by infectious disease, but by pointing out that bats live very long for their size and they don't get cancer and they regenerate tissues very quickly. So this idea of long life and bats, which has a very ancient cultural history in China, helped the founding authorities in Singapore funding him. He's a close colleague of people in Wuhan. I'll talk about that in a moment. So he was involved in describing Nipah virus outbreaks that hit horses in Australia and pigs in Malaysia. So Nipah viruses are huge problem. They're transmitted by this big flying fox, extremely cute animals, but they travel very, very far. You're all familiar with the precursors of COVID-19, SARS and MERS, a connection to bats in both cases, bats via badgers and then bats via camels that gave us MERS that went all the way to Korea as well. And probably one of the scariest of all is Ebola, which is a filovirus, which has come up again and again. It was first discovered in Yambuku, what was Zaire at the time, through a dispensary run by Belgian nuns. We're back to Belgium. Almost all these Belgian nuns died from Ebola because they were giving patients injections for vitamins with the same syringe. They essentially acted as the amplifier. And in the best, they really meant very well for their patients, but they caused a huge problem. So Ebola hides in these big fruit bats that are non-symptomatic, often don't even have antibodies against the virus, but they can deposit a lot of it when they stop at a fig tree. And then it can go into local non-human primates that die just like us. So there seems to be no difference for Ebola. Chimpanzees and gorillas die very rapidly. And when people die, then there are really important cultural connections, such as mortuary practices where you clean the body. That causes the entire family to die. You know, they go to a clinic, and then everybody at the clinic dies. The good news is that the last terrible outbreak in West Africa was incredibly well contained by these African nations of Liberia and Guinea, where they completely changed the mortuary practices. They tracked people. They used DNA sequencing to find where the virus was going. The virus did not even make it across Africa. Absolutely unbelievable. I remember in 2014, I came back from East Africa, and parents at my children's elementary school said, "Oh, you were in Africa? Weren't you afraid of Ebola?" I had to point out to them, you know, Liberia is on the other side of Africa. And by the way, I'm more likely to get shot in this elementary school in the US than to get Ebola. So sorry, that's my European... I'm a US citizen by now, so I'm allowed to say it. So things are complicated because it turns out bats themselves are experiencing climate change and a destruction of their habitat. And so some of the bats are getting completely new diseases, such as fungal infections. People, of course, who study diseases know nothing is in isolation. If you study the bats who have the fungal disease, guess what? They shed way more virus. So the bat getting sick, him or herself, is going to have an influence on how much other viruses, including COVID, that same bat is shedding. So that's why disease in animals and disease in humans is so important to follow at the same time. And you know, this is what people at the Wuhan labs did, and I'm sure you're aware of the huge acrimony going on, right? There's some very, very vested interest in pinning the pandemic. It must be the fault of A, the Chinese, B, scientists, C, you know, the Chinese scientists. And it's an irony because Dr. Zhengli Shi, who's a colleague of Lin Fa Wang, for years have been studying viruses in bats for the very reason that she was convinced something that what just happens could happen. In this paper, she predicts, her team and her predict that this is a present danger, that something like the pandemic will happen. And this paper was published just before the pandemic. So she is massively being attacked, but it's complicated, right? Because any research on viruses in any country with a big army will include a DARPA arm, or the Chinese counterpart of DARPA, which was active in Wuhan. So I haven't made up my mind, but I'm not ready to jump on, oh, the Chinese gave us the pandemic. Right? The data simply are not there. I'm not saying it's impossible. lab accidents happen. An influenza from a Russian lab did cause an influenza pandemic. So it's possible, but so far I'm not convinced by the data. It's a very ugly discussion. More importantly is the fact that we humans, including here, continue to non-sustainably harvest natural resources. I'm showing some tuna fishermen there. I love sashimi, trust me. But it's not sustainable. It's exactly the counterpart of bushmeat hunting in Africa, where you go after prized food, and then the worst thing you could do is have these live markets where you have huge density of very sick and terribly traumatized animals that are sold live, and they can, things can jump from one to the other. I'm going to round up going back to the grandmothers I briefly introduced. So I talked to you about the fact that your cell surfaces have a flavor. There is one cell there, an immune cell. You see the green around the cell. That's the flavor of the cell. Those would be carbohydrates or sugar chains. Immune cells have little protein feelers, like baseball gloves, that can touch the sugars on other cells. And they can feel other cells and decide, is this other cell healthy, or shall I call in the cavalry to kill this other cell because it's infected, it's moribund, it's cancerous, it's in the wrong place, and so forth. So this is a collection of different proteins like that. They're called siglecs because they look for sialic acids. So on one hand, you have your cells covered like a kelp forest, and on the other hand, you have another cell that can touch the kelp and ask, does it look okay or not? You remember that cartoon. So interestingly, chimpanzee cells look different from human cells because they have two different types of sugars. And this is the interaction of an immune receptor in a human. There's only one flavor, and the same immune receptor in a chimpanzee. The brain of mammals only has this one sugar. We don't know why that is. Even in chimpanzees that have two flavors of sugars on their cells, in their brain there is only one. In humans, human cells, microglia cells that are the immune cells in your cell, in your brain, they express this protein, chimpanzees much less, and that has created a situation where pathogens such as gonorrhea can actually engage. They can pretend to look like self, come up to your immune cell and say, am I not looking wonderful like you? You should tolerate me. And that's terrible because gonorrhea causes loss of fertility. Chronic gonorrhea will completely kill the fertility. And so humans have responded through evolution, and they have changed this, and they have shut off this recognition so that the pathogen cannot exploit it. And guess what? If you have this in your brain, you're protected from Alzheimer's disease. So this is work that I have done with my team and Ajit Varki's team recently, and the overall theme for that was maybe we ought to thank gonorrhea for our grandmothers, because protecting the mind of aging people is incredibly important if they are the only repository of knowledge. Until 5,000 years ago, it was only the old people who told stories. Right? And there were way more old women than old men because men are fragile and die more at every stage of life. So the fact that we have a response to a pathogen that now comes in handy to keep your minds active is it's kind of a provocative idea, but it's not new, the idea that pathogens should have contributed to who we are now. And I'll end talking about viruses. If this is indeed the case, what this means is that this is the dogma in evolutionary biology, that as you go from birth to death, the strength of natural selection decreases. When you're done reproducing, you can die, which is why most species die when they don't. But guess what? In humans, we don't die. And so the idea is that what this allows to do is that old people, especially if their cognition is protected, can contribute to the reproductive success of their young relatives. And that would be unique to humans because that contribution is linked to language and the transmission of knowledge. And possibly also happens in orcas, but they don't have language as far as we can tell. So this is the cartoon of this idea of the human lineage and the great ape lineage, and a mutation in the deep past about 2 million years ago that changed the flavor of all our cells. And then a battery of things that have changed only in humans, and very few have changed in the chimpanzees. More recently, we've done that comparing humans with our extinct relatives. Remember, we have ancient DNA from Denisovans and Neanderthals. So we can go to their genomes and ask, were they like us? Did they also have to change all these things? The answer, it looks like In some cases, yes, but in some they weren't. So there were unique differences that were different between humans and even Neanderthal and Denisovans that seemed to be involved in protecting the mind of older people. And they're expressed in a lot of different tissues, which we don't know about these extinct species because we don't have tissues. We just have teeth and bones. We cannot look at the tissue expression. So I'll end with a few comments about viruses. So a few years ago, I got to know this producer for the Korean Educational Broadcasting Service. And they contacted me because they were making a documentary with the help of a famous actor, Yoo Yun-suk, on the post-COVID. And the idea was to discuss the importance of viruses in evolution. And this was how they graphically depicted. This is the cellular tree of life with all the different cellular life forms we have, and the notion that there are all these millions of viruses that we cannot really link to the other life forms. And you've all heard that our genomes are full of junk DNA. Well, people are now starting to appreciate maybe it's not junk. So the pie diagram you have there, you see in light blue, 8% are LTR retrotransposons. These are viruses that are parked in our genome. So all of us sitting here have 8% of what defines us as human is actually thousands of viruses that we inherited in our ancestors, and they're now parked there. What do? For the most part, nothing. But in some cases, they do things. In some cases, they reawaken. For example, in amyotrophic lateral sclerosis, it is a virus like that that reactivates in your neurons. Then your immune system slams your neurons, and then you're paralyzed. So there is some of these old, distant viruses can park in you for millions of years and then rear their ugly head. So in summary, I want to remind you that it's clear, you all know that, we've become a planet-altering force. Human contact with wildlife is ever more invasive. We go to more and more places, cut things down, live there, exploit. Microbes can become pathogenic after they cross into novel species. I haven't really covered that much, but this is really the idea of a zoonosis. Even a virus that doesn't cause a bat to make antibodies, if it gets into you, it gives you full-blown Ebola and kills you in a couple of weeks. Evolutionary changes in human cell surface molecules are shared by several animal species, including some mammals and, very importantly, the birds. That is the influenza story. Wild animals can be affected by novel diseases, such as the white fungal virus in the bats, and this increases the chance of them spreading pathogens to humans. We have to stop the uncontrolled pillaging of wild animals for human consumption. That's one of the major avenues. But we also have to rethink how we use animals in the context of emergent disease, spillover of infections and antibiotic resistance in farm animals. Most farmers have noticed if you just give gratuitous antibodies to their cattle or any other livestock, they gain weight faster, just like us. Antibiotics use is correlated with weight gain. For a farmer, that's interesting. If the antibiotics are cheap, it's worth it. But what do? You breed all these resistances when you do that. So finally, we can only hope that human prosocial tendencies, which clearly exist, will allow us to take better care of each other and of the life support system of our planet. And with that, I thank you very much, and I think I'm on time.